right, so last chapter, a lot of material, but as I said before, that a lot of them are completely for informative purposes, and we will not spend much time on those because just you will see that it is written in the bottom slide, self-explanatory, and literally there is no notes in there. So if you just read the slide for the information, you will get it. All right, so the first part is the evolution, and the next part is the emergence. Is that these two topics answer these two questions: where did the virus come from, and where they are basically going. So it's interesting is that how old, I mean, like there are some retrovirus that actually appear to be found or there by these molecular clocks, which are about 450 million years old. And but we really do not actually know that when they were actually. So you can just read the descriptive part in these slides. As I said, they're very self-explanatory. So here is one of the concepts. This needs to be explained. Is that the origin of a virus, and we are still elusive. And it's kind of like a metaphor that I put at the very bottom. That is almost philosophical, that these theories are just like your particle physics and cosmology. We know a lot from Big Bang, how the origin of the universe it is, but on the other hand, we do not know anything about the dark matter energy. We just know probably it's there. So this is just like that metaphor. So here, there are three hypotheses, and let's take a look at one of them at a time. Is that first is the primordial virus world as well. In this process, what happened, that all these biochemical and macromolecule, organic molecule, they evolved and came together, and that process, they actually became selfish replicators, cellular genome, cellular genes, they just all combine into replicants, and they actually evolved into the virus. On the other hand, what happened is the escaped gene hypothesis. It started with the primitive cell or the protocell, and some of these genes actually escaped, and some of them are replication competent parts, and some of them actually produce capsid, selfish replicators, and these maybe start small and acquired more and more genes as they infected new hosts or try to new cells, that kind of thing. So this escape gene hypothesis. And the cellular regression model is basically that it was started big, like in a modern cell, and then some of these specific set of genes actually escaped them, and after that they actually became obligate intracellular parasite, and they evolved that way. So the question is that there are three hypotheses, and all of the hypotheses still holds true, because the way hypothesis works, that unless it is proven wrong, it still holds true. So that's how it is. And some of them are, all of them are supported by some of these viruses. So think about that. If you can think about the viroids or the naked RNA or the satellites, they are probably the primordial virus world. Then many small acquired, most of the viruses, this escape gene hypothesis may work very well with them. And then finally, the discovery of the giant viruses like the pithovirus, Pandora virus, Mimi, Momo, Mimu viruses, all these viruses that you know, the giant viruses, are probably due to the cellular regression. So here, these are just really recently known, last 20 years. Some of them discovered 2013, 2014. And they are from the permafrost. And the interesting part is that throughout all the viruses that we were talking about, the genome is very tightly packed. There is no empty space in the viruses. That's basically the idea. Here, some of the viruses actually contain about 2 million base pairs, 2 million base pairs of genome. Okay, all right, that's packed. But there are examples where they have about 60,000 base pairs. So here, there is a huge amount of empty space, and that gives you an idea that maybe the regression hypothesis holds true, and maybe the size of the virus did not got small enough or catch up to the smaller genome size. And at one point of time, it will. And again, remember that all these ideas are completely speculative, and but they are not proven wrong yet. So all that we can tell you about the origin of the viruses. Now, the DNA viruses, this is a very interesting thing, that how we know that how old are these retroviruses, 450 million years old. You don't get the fossils of the viruses or anything like that, but you basically get their genome fragments even with the herpes viruses over the period of time. Remember that some of the herpes viruses can cause cancer. In that case, the genome or the bits of their genome becomes a cellular part by integration. And this is not just like the integration like the retroviruses. It is just like a bad homologous or analogous recombination or a homologous recombination, or not analogous, sorry, I'm sorry for the word. Heterologous recombination, just a double-stranded break and joining of a part. And those herpes viruses, double-stranded DNA become the part of those. And those basically serve as molecular clocks. And by understanding those, or basically analyzing the genome sequences, you can easily see that about 120 million years ago, they evolved into alpha, beta, gamma, and fish, oyster, amphibians have those. And these sequences of herpes viruses actually are the signature sequences that is found in the present ones. So we can just clock back that how old they are. And mammals about 68, 80 million years ago. Now, here is this thing that probably at this time you just really believe in front of your eyes, that the public is constantly confronted the reality of the viral evolution. So you have seen HIV, West Nile, particularly in the US, hepatitis C virus, Ebola, SARS. This is the classic SARS of 2003, MERS, Zika, and the SARS-CoV-2 variants. I mean, like, Omicron is a concern 3 days ago, basically. And you can see that it started with an alpha, then from you have a beta, then you have a delta. I mean, like, I'm just not talking about all of the things. How many clades that has been passed, you can easily see that. So that gives you that how the viruses actually evolved. And then with the influenza and a common cold, every year you have a tetravalent new type of vaccine that is required because the evolution of this influenza virus, and one become the most predominant circulating strain. HIV constantly evolved as they replicate. That's the reason that you need this multiple 3 or 4 drugs so that the mutations or the resistance due to the mutations doesn't arise. So these are some of the things. And the fact is that the viruses evolve faster than many can comprehend. And I will just justify that statement what it is. And we will see that this link in there, and you can see the SARS-CoV-2 evolution in real time from the January of 2020 until today itself. And you can just follow that link to do that. We have we'll talk about that in later part as well. So here what we are looking at, that the viral, which is basically a non-living entity, evolution mimic the adaptive evolution of the host, and see that how it is. So here we are describing a host which is a living animal. Just think about a human being that evolved, how they are evolving. So here you have a host protein adapted to the environment, and then there is a change in the environment, and the selective region, the host protein maladapted to the environment. So here the protein doesn't offer any advantage to be in that environment. So you undergo adaptive evolution. That means there are some mutations that produced a different kind of protein, and the host protein adapted to new environment. And the ones which were not, these are which you call as a deleterious alleles, gradually got deleted in the population. And these are selected over the period of time, and the host proteins actually maintained in a new environment. And we have really great examples of some of these. Say, for an example, the sickle cell anemia was very predominant in Africa, but they are malaria resistant. And we see an unbelievably high number of these sickle cell anemia, heterozygous or homozygous, in the African continent. So it directly tells us that how this basically works. And the virus actually works this way, being a non-living. How? When you are providing measures, social distancing, putting on masks, have antivirals or a vaccine, the virus is not getting enough chance to infect the other. So the only ones which can infect the others becomes highly contagious, they are getting selected. And this is the selection pressure under which, so it is a constant race, and that's how just these barriers make the virus. It's not the virus is intelligently doing that. It is just out of the many that is being replicated, the one which is fit can transmit to the other, and that's basically what is happening. So basically, there are four main drivers of the evolution. It's a large number of progeny. It's a huge understanding. I mean, like, this will take millions of years for the humans. Large number of mutants and the quasi-species effect and the selection. And we are going to explain that what these are and what this is quasi-species. So if you just think about the population of a virus on the right-hand side, what it knows, that the black line is a genome of a virus, and each of these different colored things, what you are seeing, is a protein mutation at that point. If you sample a person for this SARS-CoV-2, or say a cold virus or an influenza virus, you are going to get a mixture of all this. But in reality, we do not represent it that, and that this mixture is called these quasi-species. So here, we'll come back to this effect in a second. So the evolution is guided by the RNA viruses and the DNA viruses at a different rate, and we'll explain that why it is. So the RNA-dependent RNA polymerase, this is always of viral origin. It lacks the proofreading activity. So about 10 to the power 4, 10 to the power nucleotides polymerized, there is one mutation, no proofreading. So in a 10 kb genome, the mutation is 1 in a 10 to the power 4 results in about 1 mutations per genome. So just think about that. One virus replicates into another one, should get 1 mutation. If it makes 10,000, just imagine 10,000 mutations. So this is what drives the viral evolution so fast. So the error rate is 1,000 to 10,000 greater than the host genome, what it is. But here is one of the exceptions also. The order Nidovirales, which is the largest RNA genome of all RNA viruses, contain the proofreading exonuclease we talked about many times, which is exo-N and the endonuclease endo-U. The coronavirus is one of the 4 viruses. All others are non-human viruses. They are fish viruses, other type of viruses. But coronavirus is one of the 4 families under the order Nidovirales. And the SARS, MARS, SARS-CoV-2, all of this actually code for this exo-N and endo-U. So they do have some proofreading activity to maintain that large genome. And so you will argue that, does that mean they don't evolve? Absolutely, they do. But at a slower rate, because there is a reason for that. We're going to come back to that. But they do have proofreading other than that. On the other hand, the DNA replication is not as error-prone as the RNA viruses. 10 to the power minus 6 to 10 to the power 8, and this is the mutation rate closer to the host, or the say primates, or the other eukaryotes that they infect. Because what happens, the DNA polymerases have proofreading. Most of them use the host DNA polymerase. So you have proofreading. And even the pox viruses and the herpes viruses have their own DNA polymerases, not the herpes viruses, the pox viruses have their own DNA polymerases, which use proofreading. They also code for proofreading activity of the DNA polymerases. And they generate less diversity and evolve slower than the RNA viruses. So here is a very interesting way that you can just predict. So what we are looking at, the x-axis is the genome size by the nucleotide. So see that this is 10 to the power 6, about 10 to the power 7. This is genome size, nucleotide. On the other hand, the y-axis is the mutation rate. So what is like a it is substitutions per nucleotide per cycle generations, basically. So if you look at that the smallest one is the viroid, and see the mutation rate. So smallest genome, highest. On the other hand, look at these. These are bacteria, and see the mutation rate is so low. On the other hand, if you just look at the DNA, single-stranded or the double-stranded DNA virus, see right here, they're in between 10 to the power 6 and 10 to the power 7. On the other hand, if you look at these plus, minus, and the double-stranded RNA viruses, their mutation rates are much higher, as low as 10 to the power minus 3 in some of the cases. So you can easily see that the genome size does matter. So the larger genomes, lower mutation that is tolerated, the smaller the genome, that their mutation frequency is much less. So what does that mean, basically? This means that there is an error threshold, and it is a powerful advantage for selection and survival balance and genetic fidelity and the mutation rate. And what we mean by that this is called the error threshold in a viral evolution. What happens that if a virus exceeds that error threshold, that means that it is sustaining mutations, too much mutation above the error threshold, then it's not going to survive. It will lose infectivity. On the other hand, and this is how multiple passages in the small cell culture causes the attenuated viruses that is used for the developing of the vaccine. Because it is not in the population, it's not a large, it's a very confined space, very limited number of viruses. On the other hand, below the error threshold cannot produce enough mutation to survive the selection. So that's also a thing that it will not be adapted to the host, and the host will survive, and basically the virus will not be able to infect the host. So the RNA viruses evolve close to their error threshold, whereas the DNA viruses evolve far below their error threshold. So here is one of the examples that was done in a cell culture. So this is an analog called azacitidine. It is incorporated as a C, like a base pair, like ATCG is a C, cytosine. But it templates as a T. That means that if this is azacitidine is in the chain, when it templates, it will template as a T, so there will be a G to A transition. So that's basically the mutation. So when this similar experiment, so what happens that if you do this experiment of putting this azacitidine, mutation rate among the viral progeny increases several orders in magnitude in the DNA viruses, several orders, and they still survive. But when it is done with the RNA viruses, the error frequency per genome increases only 2 to 3-fold at best. What it means that they are close to the error threshold and cannot make more mutations. So what does that mean? So let's take a look that this is what it is. So the ribavirin is one of those very old nucleoside analog. And due to that, suppose there are some mutations in the genome, and just look at this right here. If you just make these 2 mutations in there, and the infectivity from here, from 100% plus, it drops to about, say, 30%. So just about couple of mutations, 3-4 mutations, make the virus completely inactive. So this is basically the error, the effect of a nucleoside analog. So that's why it is being said that this is poliovirus. Remember that plus-strand RNA virus, picornaviridae family, they evolve close to the error threshold. If it is a herpes virus or a pox virus, it would have probably sustained more mutations, like maybe 100 mutations will not even make that virus, will not cause the virus to lose its infectivity. So that's basically the overview that how it works. Now we are going back to this, that what we talked about, that what is the quasi-species effect. So that is a very interesting definition, and I just put that whole wording in there. And this was a paper published in Cell in 1978, and this was way ahead of the concept before that. What they found is that they were looking at an RNA phage, and this is called a Q beta phage. The population is in a dynamic equilibrium with the viral mutants arising at a high rate on one hand and being strongly selected against one other. The genome of Q beta cannot be described as a defined unique structure, but rather as a weighted average of a large number of different individual sequences. What does that mean? You have a bacterial culture. You purify the qubit of phage from there, you get a sequence. Okay. That sequence piece, single piece, is just one representative sequence that has a stronger signal. There are multiple single nucleotide polymorphism in that culture that you did not pick up. And that's basically what it is. So the virus population, that what it means, exists in a dynamic distribution of non-identical but related replicon, which is called a quasi-species. And sometimes they are called the mutant swarms, mutant clouds, but the quasi-species is the right word. So what we mean by that, if a person right now, I mean, like we could just make this reference with the common cold and influenza virus, or right now for the SARS-CoV-2. If you take the viral samples from someone, in a single person, probably all of these exist. So the basic backbone is the linear representation of their genome. And all of these small things that you are seeing is a mutation somewhere there in their genome. When you take the sample and if you sequence that, you will basically get one sequence which does have the highest signal to ratio, noise ratio. That means that you will basically sequence, or the read through that you will adapt from there is that where you have the highest signal to noise ratio. But there are, in the background, there are many. If you have the ability to pick up them, you will pick up additional ones. And that's how in the population, in some places you have one less, in some places you have one more, and these quasi-species effect can be caught when you take the samples from different patients as such. But each horizontal line is a genome, and the bottom line is a representative genome. That's what we are seeing, that you are, although there are so many mutations already in there, we are just catching them at the bottom. Only when they emerge as being the predominant, we are catching. So what do we mean by this? The Omicron now suddenly becomes, last couple of days, the most predominant strains for the SARS. It was probably evolved almost 6-8 months back, even maybe earlier. And it was circulating but did not become a predominant strain, and now suddenly did that. And this is what it is. So what happens that when you have this mixture of the viruses in a single person, if you sequence, you will get this, which is just like the nucleoside sequence. So the nucleotide sequence derived from a mixture of all genomes from the left, but this represents just one representative sequence read among many on the left. So usually you pick up the most predominant ones. So why suddenly people get concerned about a new strain that is coming up? What it means that out of that mixture, suddenly Omicron became the most common one compared to the others. That's why you are picking up that, because the signal noise ratio for the Delta when it was discovered, or so Omicron, is become the predominant. So that means that it has some advantage, and that is more in the population in the left that what you are seeing. And that people assume that going to be some kind of selection advantage that they have. So that's basically the complete understanding that what we mean by this quasi-species effect and how this happens. So here is some idea that how what happens. The initial population, you have the mutations that improve their replication, increase replicative fitness, large population passages. This is exactly what is happening in any of the epidemic, pandemic, whatsoever, in the population right here. On the other hand, if you just take just one of these and passage them, say in a cell culture, you are creating a bottleneck, and this is basically a small population passages. So you just take just one. Here what is happening that in the population, all of these are being transmitted to a new person, then a new person, then a new person, all of them. But you are doing a plaque purification. You are just taking just one of them, one clone, and then putting them in a cell culture. Now what you have repeated bottlenecks, accumulation of mutations, decreased replicative thickness. And this we always see that in the culture over the period of time, this is how the attenuated strains basically develop. So The viral diversity is essential for the viral survival, and in the current pandemic, being so many subjects out there to infect, the virus is really super happy to do that. When there is not many person that it can infect, then you create a bottleneck by the herd immunity, and the virus will gradually become endemic or gradually disappear. That's how basically how it works. So here is another rendition of the genetic bottleneck. You have a parent population. See, bottleneck is a drastic reduction in the population of the virus. Remember that, drastic reduction in the population. This is the surviving generation, and now you have the next generation. So here, see that from predominantly there were yellow and the blue were in equal amount, and here gradually in the next generation, the blue is selected because initially with the bottleneck, you selected more blues. And this is basically can be done by this plaque purification. And what happened that gradually that may lead to less fitness of the virus. This exactly works in a large population. We know that in an inbred population, disease exists. In cats, in dogs, when you have these champion bloodlines and all those kind of things, when they're inbred, they do sustain these mutations or the genetic bottleneck, which are disadvantageous for them, and they stay. This is basically the basic law of eugenics that also happens in the viral evolution. So this is a metaphor, is like the ratchet metaphor. So each new mutation in a virus works like a ratchet, allowing the gear to move forward. It cannot go backward. You have a mutation, and then reversal of the mutation to go back to its original genome is very unlikely. If you are evolving, you are always moving forward. And each round of error-prone replication works like a ratchet, clicking relentlessly as mutations accumulate at every replication cycle. And this is the most interesting part. So for the human being, this is one generation, one person to another. And this replication cycle is just a mere couple of hours. And think about the number of progeny versus how much a primate can produce in one generation. So this is the idea. So what happens is that what are the bottlenecks in the real world? So it's a small droplet suspended virus aerosol transmission, activation of the latent virus from a limited population, small volume of inoculum introduced by the infection of the insect bites. These all basically cause the bottleneck, and that should take care of most of the viruses. And that's why we are not every day succumbing to many of the viruses. But what is happening that the question is that, okay, all right, so if we're seeing that, okay, we are just breathing a small droplet from a person in a SARS-CoV-2 as well, and why for 2 years this is what it is. The reason is that already there are so many people infected with that although there are some genetic bottlenecks, the ones which are evolving to be more contagious is selected in the population and spreading. So that's basically what it is. And that's the reason that we are not having an Ebola, Marburg, or that kind of viruses around. Or say, for example, the influenza rates are also low, although they are very infectious. And because of these genetic bottlenecks, the number of people that are carrying and transmitting are less. So this is basically the idea that how do infections that spread by these routes escape the Muller's ratchet. This is the process that I said, is that when you have much large subject to transmit the virus, each of these droplets that they are transmitting from different person, it contains different viruses. And in the larger population, it is easy for them to evolve. So that's why you are going to see that over the period of time that you're going to see a peak of this virus increasing and decreasing, increasing, decreasing. Each time there is a new variety of the virus is doing that. And gradually this peak will probably become shorter and shorter over the period of time as the number of subjects due to infection or vaccine or measures actually decline over the period of time. So this is very relevant for the understanding in terms of the COVID-19. So here, avoiding the ratchet, another strategy. Say, for example, this is a retroviral strategy. So think about that. The retrovirus contains two identical copies of the genome. It's a very error-prone, like but the RT or the reverse transcriptase is error-prone as well. So here, the sick virus 1, which is the red line, and here there is a mutation. And then you have a diploid copy of the same genome. So the red is one copy of the genome. This red is another copy of the genome. And this is a sick virus 2. Here you have mutations, different places. But remember that the retrovirus genome actually uses the copy choice mechanism. And this is just by chance. The virus doesn't know which one to copy. But if the copy choice mechanism goes like this, switch the template and copy this, then you have one functional genome, which is healthy viral recombinant. This will basically get selected. Now, how this virus knows? Now, virus doesn't know. It will make a lot of these sick virus 1 and 2 as well. Plenty of them. They will just get sick, they will just not transmit, and they will not just get selected. And the healthy viral recombinant will just get selected. So this is basically that one of the process that how you avoid the constant mutation pressure, which can render the virus ineffective, or stop the transmission. So here is a selection of the influenza virus, and there are two ways that they can do that. One is what we call as a genetic drift. The genetic drift, and very important for your quiz and exam questions, know the genetic drift and the genetic shift. The genetic drift is actually a small change, like mutations every replication cycle, which changes some of the specific amino acid. And here, this is just the structure of the hemagglutinin HA or H. On the other hand, you know that the influenza virus is a negative strand, but it's segmented RNA virus. It contains 8 copies of negative stranded RNA genomes. And what can happen that at the same time, if one cell is infected by 2 strains of the influenza virus, during the packaging, they can mix and match. And that is a genetic shift. So basically what you are seeing right here, H1N1, and then you have an H2N2, or originally they are all, reservoir is the water birds, ducks, and waterfowls. And now you can see here, this one, and this one, HA and NA, they are from the H2N2, but all the red ones came from the H1. On the other hand, now let's take it to the H3N2. Here, you have an HA right here. The NA is from here, N2, and whereas the HA is from this H3. So this, you can see that each one of them was a Spanish flu, Asian flu of 1957, a 1968 Hong Kong flu. All of them were pandemics or epidemics, and all of them arose for this sudden shift by reassortment. And this is still ongoing, and that is the reason that each year the surveillance has been done. We saw that how the vaccine is developed and how the WHO actually make the surveillance or do the surveillance of each of the top 4 circulating strains for that year, and we have to do this all the time. Will we have to do that for the SARS-CoV-2? We'll see. We don't know. So here is a funny experiment that they did. I thought that this is something funny. Just know for the information purpose. So these European rabbits were introduced in Australia in 1859. And they actually reproduce like fire. And basically, the vegetations after vegetations were like gone by these viruses. They could not do that. And this was a national disaster. And what they did, this 1950, unfortunately, they actually got this rabbit pox virus, which is a paramyx severity negative strand RNA virus, were introduced deliberately to release the control of the population. And it was 99% lethal for these rabbits. So they killed all these rabbits. Within a 3-year period, it was only 30% effective, and the rapid reproduction outpaced mortality. It did not work. So basically what happens is that the virus evolved to kill fewer rabbits and extend the life of lethally infected rabbits so that the virus could overwinter and spread in the spread mosquitoes. So this is also the thing. If the virus kills all the subject, it will not be able to sustain itself in the population. So it's going to disappear. So it's going to balance, okay, all right, I'm going to kill, but not too many, or whatever the balance is. And this is not that it is done by an intelligent process. It is just that how the evolution works. It becomes, the host becomes an equilibrium with the pathogen. This is going to happen with the current pandemic, I guess, as well over the period of time. So here, if we just ask that is the selection of positive or a negative trait, it's actually both. So the positive trait, increase the virulence, is a consequence of high viral loads. So you make more viruses, and this will facilitate the transmission. More viruses means it is easier to transmit the virus. On the other hand, a negative trait, increased virulence reduces transmissibility because the host die faster, reducing the exposure to the uninfected host. So that's the reason that the viruses always find a balance in between the positive trait and the negative trait. Well, high viral load, but again, probably lethal, but not to some extent. That's the balance in between. So here is another thing that how we look at these arms race within the virus and the host. So suppose this is a receptor. This takes much time. This is not going to work in a context of the current pandemic because the time is too slow. This happens for thousands and thousands of years, if not million. So here you have a virus which binds to a receptor, and then basically the virus at that time, the host is basically winning in the sense is that in this case, the virus binds to an antibody and it can detect it, and the viral actually evolve in there. It can escape the antibody and the virus is winning. And now again, the antibody can bind to the virus, and here host is winning over the period of time. So we are just having this constantly, and this is what we are seeing in the current condition. Now, instead of an antibody, this could be a receptor as well. Remember that this is just a host protein, and here is an example of an antibody. It could be any host protein examples. So here we are looking at the evolution-guided functional analysis, and there are three phases of this. So let's take a look that this is a primate phylogeny, and this is just a nucleotide sequence of a certain protein, just like say protein 1, protein A, or protein X, doesn't matter. The red ones are basically non-synonymous changes in codon. That means that they will produce amino acid which is different than what their parental one was. Whereas the green codon are the synonymous, and they are changed. That see that they are always at the third place. That means that they are going to produce the same amino acid. So they're mutations, but the same amino acid. So what we basically tell that how it is expressed, if you have a synonymous mutation, we call them as a DS, and D like a small d, DS. And the non-synonymous, on the other hand, is called a DN. So the ratio of this DS over DN, this basically is determining that what is going to get selected. So here are positively selected lineages, and these are the positively selected codons. Just one example out of all these, remember that. So now let's take a look at that what they are basically look like. The second phase is that we determine the phenotypic consequence of this ortholog variation. What do you mean by that? This is the same, all of them are same proteins, and that's why we now call them as an orthologs of each protein. So in these cases, if this protein is, in these cases, the virus is basically winning. In these cases, the host is winning. Now remember that there are some of the silent mutations where the host is a winner. But in some of the cases where the host, the mutated form is not advantageous, but here it is, in this one it is, in this one it is. So there is always some advantages of the mutations. In some cases, there is not so much advantages of the mutations in the host. It's an equilibrium. Now what happens that this is how you can map the positively selected changes onto the host virus interaction. Here you can easily see that in this one, the virus is winning. In this two, this one and this one, the host is winning. Remember that some of them can be silent mutations, some of them can be ortholog variant, and right here, what we are looking at, here you have a serine right here, whereas in these two, where the host is winning is a glycine to glycine. Now what you do that you just take this one, and if you just replace this serine with a glycine, you can just tell that the host will win in this one as well. So this is how you can map a protein and look at that how the virus, some of these mammalian proteins evolved under the selection pressure of this virus infection. And this is an unbelievably classic example of that. This is a transferrin receptor 1. And what you are looking at, this one is a transferrin receptor right here, and each one of them right here is the amino acids which are from the human being. And here you are looking at basically the structure. It's the basic structure is a human TFR dimer, and it was observed in the rodents basically, but the structure is from the human. But again, they're really close, so it doesn't matter. Now, what we are seeing at that, this is the transferrin binding region, which basically takes up the iron and delivers it in the cell. So this is the region where is the active site for the iron binding site. But see the outside, these are the areas which were exploited by the arenavirus, and this is a mammary tumor virus, basically, murine mammary tumor virus. They used to bind in these regions. So arenavirus for the gray and the blue. Now, these are some specific mutations right here. These are some of the specific mutations that these are right there. That's being observed, and what you are seeing that you have these mutations where the protein adapted so that it is not infected by this arenavirus or this MMTV, whereas there were no mutations in the core part. So the activity of the protein remained, but the part, it allowed these mutations in parts where the activity is not directly involved. So this is how just over the period of time, and this thinks about that from a mouse to human, I mean, like it takes a very long time to evolve even in that process. And this just gives you that the two virus host races, how it can be determined, that these mutations basically make the virus ineffective or it cannot bind. And this is seen in this structural studies. So that's the bottom line of this. So from here to here, it took 8 million years. From a virus, say poliovirus is through gut, you can do it in 5 days. So it's the same thing. And that's where, like say for SARS-CoV-2, this number of evolutions, a person is shedding the virus, it can be within 7 to 14 days. So you really, you just have to imagine that this is basically what is driving this evolution. That is the most common thing. I mean, like you will be able to appreciate after this class that the data which is out there, and you will be able to answer a lot of questions. I can believe that if you're not in a virology class, just by biology, you will not be able to answer this many questions about SARS-CoV-2, what's going on, and what you will get after this class, particularly this one. All right, so emerging viruses. This one was in 2003, though. This is not for SARS-CoV-2. This is for the original SARS. Then the bird flu came, more of them, H1N1. And this one is from earlier as well. And the bottom ones are from the current. So you can just easily see, I mean, like just read the material. And it was kind of an interesting that I put it somewhere in the writing is that after the 2003 SARS pandemic, there was a meeting in the American Society of Virology, And Susan Weissman, she is a very known coronavirologist from the UPenn, University of Pennsylvania. They have a very strong task force of SARS-CoV-2 as well, got a lot of grants. She just told, there was a slide that she just told is the next pandemic is going to be from a coronavirus. Just that was one slide right there. And I mean, like, it's very difficult for people to just put that in there. But I saw that slide with my own eyes like 15 years ago. And that's kind of like really mind-boggling that some people can predict. But, well, so this part, we start with this emerging viruses, and the rest is all very descriptive, so you can just read that. So the emerging viruses is an expanded host range with an increase in disease not previously obvious. And many of them, the virus comes from animal to human, and this is called a zoonosis. And the infection is called a zoonotic infection. So that's a very important word for that. Remember that a zoonotic infection where the infection comes from an animal to the human being. And also the cross-species infections from SIV, simian immunodeficiency virus, moving from chimps to human and become HIV. This is another way that emerging viruses can develop. And often the cross-species infection cannot be sustained. That's also the case. Not the case for the SARS, but for the, or say MERS or the others, that's not there. Definitely Y SARS and original SARS, the 2003 SARS or the MERS, is not that common because the infection cannot be sustained. So each time it has to come from the original reservoir to the human population. Each time has to be a new spillover from the host. On the other hand, same thing for the Ebola or the Marburg, each time it has to spill over from a bat or something. On the other hand, what happens is the SARS-CoV-2 is perfectly fine for the human-to-human transmission. HIV, the same way. And that's how they want this process, although initially they were zoonotic infections. So the role of basically the evolution in emergence that leads to the biodiversity of the pathogen existing in nature, which we call as quasi-species, and adaptation to new host and the environment through variation and selection. And more transmissibility that we are seeing, at least for the Delta, Omicron we do not know yet, but that definitely the more infectious is because it undergone the selection pressure due to the measures that the humans are taking. So this is an overview of this slide, so just understand that. So we are looking at first a stable. This maintains the virus in the ecosystem, just like, say, for an example, your cold virus, influenza virus, these viruses, stable, maintain virus in the ecosystem. And the resistant host block the infection completely. The human being doesn't get infected, say, for an example, vesicular stomatitis virus. They are animal viruses, not human viruses. Blocks the infection completely. On the other hand, evolving is the instability and predictability of viral passage from experienced to naive population. And that's basically is what we are seeing with the SARS-CoV-2. On the other hand, the dead end is the one-way passage known as spillover, non-efficient transmission of the viral emergence. So this is Ebola, Marburg, we talked about that. And even the SARS or the MERS, original SARS, and the MERS, Middle Eastern Respiratory Syndrome, those are the dead-end viruses because they actually, the civet cat was the origin for this intermediate host for the SARS, and the dromedary camels, the Arabian camels are the intermediate host for the MERS, and each time an infection probably has to come and spill over from them. Otherwise, it will not sustain in the population and gradually go away. That does not mean that it is not infectious from the human to human being, but they cannot sustain that for a long period of time. And that's basically, so all of these actually stay in an equilibrium. So here is a very interesting life cycle of the arbovirus, which is the West Nile virus right here. So you can see that they can replicate in different wild birds, chickens, as well as they can replicate, just not vector, in the mosquitoes. They don't cause disease, apparently any disease in this. And the horses and the humans, they are just incidental hosts. If the mosquito bite, they get transmitted. So here you can see that right here is a stable host virus interaction where the virus replicates in them but doesn't cause any disease. Very, very interesting way that this how it is. Here is an example of, say for an example, the emerging new host, say SIV1 from the chimpanzees, and that can be transmitted to the human beings. And the problem that have, the rare chance encounter of viruses with a new host may never be detected. And that's the same case, we could detect it for the SARS and the MERS, but not probably for the SARS-CoV-2 yet. And the single host infections are transmitted to humans for many reasons for this. So this part is very descriptive, just a lot of information. So you can easily see paramyxovirus. Bat is one of the reservoir for that, and severe disease in domestic animals, you can easily see. Infect the humans, stable reservoir for coronavirus in bats and birds. Then this is a Nipah virus outbreak in Malaysia in 1998. And these are some of the bats, fruit bats, basically they are the carriers for that. And the date palm sap contaminated by bats, that actually started it in India and Bangladesh. And the human-human transmission infections continue, but it's at a very low level. This is one of the emerging viruses as well. Then Hendra, this is in Australia, 1994, racehorses. There is a vaccine for this. This is another paramyxovirus emerging viruses. And then you have these hantaviruses that actually appeared in these 4 states of the New Mexico in 1993. And it's kind of funny that the scientists actually wanted to name that as a Muirtocanian virus, and the local people actually objected. You know that why it is, right? Nobody wants to tell where they live, like I live in Death Valley. Maybe this is okay, but if he says like I live in Death Valley, town of Death Valley, or Death Canyon, weird. So this was funny, I found. So from the rodent excreta, this is, so hantavirus is still, there was some infections from the rodent excreta in the Yellowstone National Park. So campers are at risk for this one as well, but not very common. This is also a Bunyaviridi family, and this is a flipovirus. And the 2012, this virus was found, and 40 subsequent cases. This Lone Star tick is a vector, and just think about that. I mean, like how a virus is being discovered, infecting a human being. It's just a chance. A person goes with a fever, fatigue, diarrhea, low platelets, WBCs. It's like you must have some chronic disease, they will get vitamin, do this, do that, and then suddenly you find a virus. So that's why sometimes it can go completely undetected. So it's not easy to understand what we meant by right here. This was thing. Single host infections may not transmitted among the human beings and sometimes never be detected as well. So it's not very easy to identify that. So talked about Ebola all the time. And 1976, it's in the Democratic Republic of Congo. That was the first, followed by Sudan, and among the cotton factory workers. Now, the interesting part of the Ebola is right here, the outbreak in 2014, 2016, infected more than 26,000, about 11,000 plus death. This is the time, so this was already in there, and there used to be sporadic outbreaks of that. But the Western world never actually invested any money in that because you know that how much time it takes to develop a vaccine. Only when a large population started to get infected, the companies told that they probably could run a clinical trial and do that. And these are the something, like if you have a small severe thing develops, it is not easy to run a clinical trial and it's very difficult. And then after this, the recombinant vesicular stomatitis virus, ZBOF, this is from Merck. The vaccine was approved in 2019. So you can see that pretty much within about 3 years after that, they were approved. So it is not that we are developing vaccines for a long time. So they are pretty good as well. All right, high mortality. Then the filovirus and the Marburg has been isolated from cave-dwelling food bats. Ebola virus RNA antibodies found in these tree-roosting bats, but not infectious virus. So we really do not know. And the human, gorilla, chimpanzees are the dead-end hosts. So each time there's a new spillover from these bats, probably. Here is just an overview of some of these zoonotic reservoirs. We already know that the SARS is palm civet cats, dromedary camels in Mars. SARS-CoV-2, some people were indicating pangolin, but it is really, we do not know. It's really, here you can just follow. This is the link towards the Johns Hopkins COVID-19 dashboard. You can see that how many in the recent times in real time from there. All right, so here, this is just a couple of slides about the original SARS, that's 2002, 2003. And dry calf shortness, 10-20% require mechanical ventilation. And this is how it started. A Chinese doctor who treated patients traveled to Hong Kong and stayed in this Metropole Hotel. And he died on 22nd of February. And the infection spread to 10 people of the hotel who flew to Singapore, Vietnam, and Canada, and spread to 29%, about 10% mortality. So very interesting way that what it is. But they actually can, human sera before the SARS outbreak do not contain antibodies to SARS. But in the Guangdong district's cases were handlers of animal of exotic food, and that's how it was found that the civet cats were the thing. So from bats to the civet cats, to the animals. So that's what came from an exotic meat market. The next one was the MERS, Middle Eastern Respiratory Syndrome, 2012, 60-year-old male patient died of pneumonia, renal failure. That was the index case in 2012. And this is interesting that the MERS, being a coronavirus, it uses a different receptor. It doesn't use an ACE2, it's use a dipeptidyl peptidase 4 receptor, DPP4. And still there are sporadic cases, but this is way more virulent, probably the more virulent even SARS-CoV-2. Less cases, much more, about 30% mortality, very, very high mortality. So that's spread to dromedary camels. Probably it came from bird infected and just an intermediate host as the dromedary camels. And there are lots of unanswered questions as well. The camel vaccine is in clinical trials and none in the market even after 8 years, no antivirals as such. And these are probably 2,400 cases, 827 deaths about the MERS transmission and overview of that. So here the SARS-CoV-2 genome is almost similar to, you already know that 96% to an RATG13, which was isolated at 2013. This is the closest one that we have. It's a beta coronavirus. And after that, how it evolved, we really do not know that how it did. And there are lots of hypotheses and theories in there. But the main idea is that when you do this global sampling, you get a direct proof of the quasi-species concept that last 2 years, how much it actually evolved. So think about that you have a bat virus which is about discovered in 2013. Maybe around that time or maybe earlier, it infected some other animals in there. And then because what the scientists say is that it is they do not think that it directly a bat virus could infect the human being. They're too distant. It will not be fit. So it was definitely had some intermediate animals where they evolved and then could transmit to the human being, but it's also speculative. So that's the idea. And the isolate just jumped to be just happened to be collected. It may exist in nature long before. And it had a chance to evolve for 6 years before it accumulated the mutations to infect humans. How? Still a mystery. This is minimum 6 years. But what we are trying to say that if RATG13 is discovered in 2013, but it may be infecting bats long before. So it evolved somewhere in some other intermediate animals for long before. That's what it is. And then I'm not going into the theory of lab escape and all those kind of things. There is always a possibility. Thus, just developing a virus on RATG13 artificially to infect that I don't buy. So I have a strong opinion about that. And the lab escape and the other things, there are very small chances of that. The reason that is, I believe it in a little bit different way. That I believe that if there is something in there, someone somehow is going to go somewhere, ask for $1 trillion and leak that. So I believe that. So if there is something, they cannot shut it down completely. If there is a conspiracy going on, someone, let's just think about that. I mean, like a person from the lab even can be like a research assistant. Will travel to the United States and go in there was like, I have a data, just give me asylum, give me $1 trillion, I will tell you the truth. So somebody just due to that, this is, again, this looks like a kind of a rogue comment, but we have seen over the period of time, this is how things get leaked. So, but who knows? So, but follow this article and have your own opinion. Everybody will have their different opinions about this. So let's go to the real material in here, that what is the current condition, that's rather than speculating that what happened, because many things can happen. So the Omicron,
25 mutations on the spike,
significantly greater number of mutations than Delta found in several countries. More mutation may sustain the virus and take over Delta, but exactly the opposite may happen. The mutations may not be able to sustain the virus as a major variant like Delta, but we do not know yet, so only the time will tell. Now, there is no credible info yet, will take about 2 weeks. What are the questions? More infectious, contagious? We do not know. Escape vaccine efficacy, breakthrough or severe? We do not know, but I will argue against this to some extent. Then severity of the disease, do know, will it take the variant? We do not know. So if you want to, so this data is directly from the WHO website, which is on the left-hand side. And you can also go to this Nature, this is our article. And here I'm just going to this and show you that this is from, all right, so this is your Delta. So when you go to the Delta using this strain, it will tell you exactly these are the single nucleotide polymorphism, all of them on that. And you see that the S, 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 so you have 1, 2, 3, 1, 2, 3, 4, 5. So 5 of them are on the spike protein. The N are the nucleocapsid protein, M on the matrix protein, and they are ORF, these are the ORF7A and the 3A, these are the accessory proteins. So these are the mutations. So you have on spike 1, 2, 3, 4, 5. All right. So this is your Delta. Now let's go to Omicron. It's developed from an alpha lineage, very, very different. Now what you have here, this is the number of mutations. So just think about that right here. And if we just look at the S, how many? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25. So 25 mutations in there. So 30 is not correct. Total number of mutations, of course, more than 30, but 25 of them on the spike. Now, remember that the most important thing to understand, all those mutations are not a single virus. This is the most important. Those are coming from the quasi-species effect. So what it means, you have isolated one from Australia. Out of all those 30, maybe 4 of them are in that Australian variety. You have one from Botswana, maybe 5 of them which are different from there. South Africa, there. So each where this quasi-species of the Delta strain is being isolated, and they are having this backbone with these single nucleotide polymorphism. It is not in, all the mutations are not in one virus. That is the most important thing. So here, what's the most important part, that you can see that over the period of time, see that how the Delta almost took over, became 100%. where the alpha right here is right, you can see this, basically it's declining and then rising. And then what you are looking at, the Omicron basically is a derivative of the alpha strain. And these are the different places where they have been spotted. And this is where you will find this interesting. This is your delta spike. So all the quasi-species single nucleotide polymorphisms, they are mapped out to the spike protein. Remember that it's a trimer. This is the trimer. Stalk, the top portion is the RBD, or the receptor binding domain, to S2. This yellowish part is just one of the receptor binding domain of the 3. So here is the second one, there is a third one, 1, 2, and 3. So receptor binding domain. And here you can see that all the relevant mutations, basically. And this is what is more important. Remember that, as I said, that they are not all in all of the viruses. So if you see 70% of the quasi-species, red ones are there in more than 70%. If you see that 1 to 5%, they are in the blue, they are in the green. So this is the number of mutations that we see more. Now look at the number of mutations in Omicron, which is the B1.1.529. And most of them are in the RBD, or the receptor binding domain. So this gives me, makes me make a different comment, and it's a very premature and completely speculative comment. It may even find a different receptor to bind with that many mutations. So I mean, like, again, this is not a scare tactic or tell something irresponsibility. Somebody, probably hundreds of labs, already jumped into doing that, whether it's binding to somewhere it is, because it's too many mutations in the receptor binding domain. It still can probably, even with that, bind to ACE2, but there's a possibility that it can do, and someone will do these works for sure. So here is the important part. A high number of mutations does not automatically mean that these variants are more dangerous. That's the most important part. And because it may not sustain, we'll see. So here is the last part that what we're going to see is that why you should not be too scared about the new variant that is coming out. And this will give you that when people say that the vaccine works, and what is the rationale behind that? Everybody tells it works. It's like, okay, all right, I'm not going to believe, so believe the science that how it is proven that it works. So here what you're looking at, the A panel, this is the IgG response against the peptides derived from the spike protein. So this is from the S1 all the way up to the whole structure, S1 subunit, which contains the 3 receptor binding domain. And this is the stalk portion. So S1 is the top, S2 is the bottom part, and the fusion peptide is in the middle, S2 subunit. If you look at the hotspot of the IgG response, where do you see which part? Do you see more red in S1 or do you see more in S2? So in this one, so this is the linear. This is S1 on the left, S2 is on the right. So where do you see more red? You see in S2, right? S2 is the stalk. Let's take a look at so this is the stalk, this is the stalk. Mutations are mostly in the S1 region, and the stalk is S2 with much less mutations, less frequency as well. So the antibodies which are going to bind in this region will still bind. If they cannot bind in here, that is okay. The efficacy may be a little bit reduced, and that is what this paper tells you, that right here is the receptor binding domain, right here, where the maximum number of mutations are. And okay, all right, maximum number of mutations, and these top ones are the epitopes from that region. These actually will develop the best antibodies for the identification. But what happens that if you just look at, this is the S1, this is the S2 region right here, and you have all these regions with very high epitopes and antibodies, and this is all in the S2 region. So if you just look at that what is being told is like, if you just look at the top portion versus the bottom portion, This is one of the hot region. This is one of the hot region. And these hot regions means that they are in the stalk portion. They are in the stalk portion of the spike protein. And they have an extremely good signal to noise ratio. That means this region basically is presenting an epitope for raising more and more antibodies. That's what it is. So it is more binding to those regions compared to the ACE, because ACE is constantly changing and they have different ones. That's what it is. And you can see that validation of the enrich from the fluorescence data. Now here is just a couple of examples of these antibody neutralization assays. Remember that this is not the complete one. So they have taken this S1 to 82 region, 1993, S1 to 105, and up to 103. And if you just look at these data, you can see that is 113%, 94, 49, 63, 105. These are the areas where it is neutralizing. Great. This is the S1 region, more mutations, but less epitope. You have these values that if this neutralization means that if you have an antibody against these epitope, which is like, say, for example, right here, S1 82, you can control the virus 94% with that antibody. On the other hand, just look at this one. If you have an S222 or S278, S222, you can control the virus up to the 110%. Of course, this is experimental. So 94 versus 110. So just think about that if you have an antibody binding in there, it will completely neutralize the virus. And see that S2 region are less mutations and more epitopes for antibodies. And that's the reason that the virus it will still work. The vaccines, what you have, and many are on the horizon. So if somebody argues with you, take this data and tell it to them that this is the science. It is that the problem is that it is very difficult, even if you're a non-virologist, to bring up this and tell exactly why they are saying that the vaccines will still work. So here is the real data. You can just follow that. The Moderna Pfizer, the efficacy for the lowered 95% chance of hospitalization to 80% for the variants. And the virus efficacy lowered to 80% to about 50 to 70% for breakthrough infection. Now remember that breakthrough infection means that somebody is testing positive. That does not mean that they will have the disease. Remember that we are happy with the 60% efficacy of the influenza vaccine that we take every year. It's about 60-65% efficacy. So still, this is good. But probably we will have a multivalent vaccine like influenza, and all the variants addressed must need a reformulative new variant arise like influenza, and all vaccine manufacturers are either developing or doing the clinical trial for the vaccine. So if you just go to the Moderna and the Pfizer website, the Pfizer, both of them are working clinically trialing the Delta variant from June onwards. So all they have to do, 2 different mRNAs, put in the same thing, just put it in there. It is really easy. It just takes 90 days for the company to do that from that to the market. They're just waiting for the data to be presented in there. And so there is very much hope that they will be able to do this just like the influenza virus, and that will take care of it. All right.